Good afternoon, everyone. My name is Jorge Villa. I am going to be the host for today. We're very pleased to have Timnit Kefela. She is a PhD student at University of California, Santa Barbara. She has a biology bachelor's and a master's from Rutgers University, and her research focuses on understanding the sources, pathways, and fates of plastic in soils and marine environments. And again, we're very happy to have her here sharing part of her research, or at least a big content of her research with us today. And without further ado, the microphone is all yours, Sydney.
Thank you so much. Something that I spend a lot of time interfacing with and studying, and that's specifically microplastic pollution. How I'm gearing my presentation today is towards, providing a state of the science of microplastic pollution, primarily in the two matrices that I study, which is soils and sediments. So I do hope to talk about the sources, the pathways, and the fates of microplastics in these systems, and I really do look forward to digging in with you all about this. All right, let's get started. So we'll start with just a very simple question. The reason why I'm asking this question is because I want us to envision the positionality of plastics in our built environment, right? So you all may be in your offices, your homes, a seminar room watching this, and I want you to think about your physical environment right now. And I'm going to ask you a question if you would like to answer this in the chat. But how many items that contain plastic can you count around you? And I'll give you about 10 seconds to respond to this. So there's quite a few, I know right around me. I have about maybe 5 to 10 items, and I'm in a tiny little office. And it makes a lot of sense that there's plastics everywhere, because plastics are such an important facet of our modern lives. They are the vessels that carry our food, our water, such as plastic bottles. They carry our fuel, for example, such as jerry cans. This illustration just shows you how ubiquitous they are. They help transport us, such as tires on cars and buses. They support our homes, such as piping. They help us capture food, such as nets and fishing lines. They entertain us, such as vinyl records, clothe us. They help care for us, such as their continuous use in medical devices, the masks in a time of a pandemic, gloves, tools, among other things. And the reason why they have such ubiquitous uses is because plastics are malleable, they're durable, they're cost-effective in comparison to their non-plastic counterparts, provided you add the right additives that allow them to have the desired properties you need. So the fact that plastics are such an integral part of our daily lives, it is no surprise at all, no surprise at all, that we have produced 8.3 billion metric tons of it since their commercialization in 1950. Looking at this vast amount of source material, it is also no surprise that we see them everywhere, because they've been found in the depths of the deepest part of our planet as we know it right now, which is the Mariana Trench, and the highest point of our planet. They've also been found near Mount Everest. So plastics have become such a ubiquitous part of our environment. But how do they become micro? Well, they can become micro with anthropogenic intentionality, or because of them being in the environment. But first, let's define what a microplastic is. So they're essentially just smaller plastics that are less than 5 millimeters in size. So we're talking smaller than the width of a number 2 pencil. One thing to note about them is that just like how we talked about plastics being in nearly everything, it is no surprise, again, that they range widely in shape, type, and size. So microplastics are categorized as primary or secondary. Primary microplastics are intentionally made to be less than 5 millimeters in size. Some examples of this include glitter, nurdles, which are the starter plastic resin pellets, which are the starter plastic resin pellets before you say a plastic undergoes some kind of extrusion to form a desired plastic product. as well as microbeads, which despite the 2015 Microbead Free Waters Act, there are still some products such as detergents that are exempted from that law and are often used as kind of like an abrasive or exfoliant. And then we have secondary microplastics, which are microplastics that form as a result of different processes. So plastic items that have been exposed to ultraviolet radiation undergo photooxidation, which results in the embrittlement of that particular plastic. That combined with, say, the force of a wave and wind can cause the brittle plastic to fragment further. However, outside the realm of these natural processes, secondary microplastics can be produced through machinations and use. And some great examples are that of wandering clothes. Wandering clothes and textiles have been widely identified to be a significant generator of synthetic microfibers released in the environment. So much so, a study my colleagues and I conducted identified that approximately 5.6 million metric tons of microfibers have been released into the environment since 1950, of which half of that has been within the last decade. And so annually, we would say about 343.7 kilotons of microfibers are released annually, at least at the global level, which to some may not seem like much, but those fibers are incredibly light. And if we were to convert them to counts, we're talking microfibers at the magnitude of trillions. So that is definitely one way that secondary microplastics can be formed as a result of our use. Another way is driving a vehicle. So driving a vehicle leaves behind tire particles, so much so that 0.2 to 5.5 kilograms per capita of tire particles are released into our environment annually. And similar to the whole point of that I brought up, that may seem like little, considering our global population, it's not. As well as if we were to convert that magnitude to counts, it's pretty high. Now that we know, why they are micro, the question is, how do they move into and around the environment? Well, there are certain factors about microplastics that do influence how they move through different environmental matrices. So one of them is size. In waters, smaller microplastics, specifically the ones that fall under the nano range, which is below 0.1 microns, they settle out faster due to higher diffusivity, resulting in an increased rate of contact, and in turn aggregation, which aids in them reaching their sinking density. So this is one way that microplastics end up reaching sediments, provided they are not ingested by organisms on the way down. In soils, some microplastics may be larger than the pore sizes of their soils that they're in, for example, vertisols, right? And they're retained in the soil's top layer. However, if they're smaller than the pore size throat, they may move further down. In other instances, however, it could be that these microplastics absorb onto soil particles and form aggregates, heteroaggregates, making them immobile, such as that with clay, proving them like low risk, for example, to say groundwater contamination, but could be problematic for, say, plant root systems. And I'll explain why that is an issue when we reach the effect portion of this talk. Another physicochemical property that influences their movement is morphology. So microplastics floating at the surface influence the rate at which they undergo biofouling triggered sinking. So for example, for spherical microplastics, those have a minimum surface area. So in comparison to, say, their foam-like and fiber-like microplastics, which influences the rate at which biofouling occurs, which further results in sinking. So this is another way for them to enter sediments. Due to the more surface area available with, say, fiber-like microplastics, the amount of time that it floats on the upper layer of the water column is going to be considerably smaller, and it's shorter, not smaller, in comparison to, say, a spherical microplastic. In soils, spherical microplastics are most likely mimic. So much of how we actually understand this is inferring this from like colloidal studies that have used like these spherical plastics and very uniform quartz sand, which isn't necessarily what soil is like, right? But based on that and leveraging the knowledge from that, spherical microplastics would most likely mimic the movement of, say, spherical colloids through porous media. And it would yield, however, it would yield somewhat different effects with fiber microplastics. So inferring this research from like rod-shaped polystyrene nanoplastics, for example, is that with rod-shaped polystyrene nanoplastics, they had a lower deposition rate in comparison to, say, their spherical counterparts in a colloid study, a colloid transport study that was done in 2013. And this is largely because the rod-shaped nanoplastics had a higher aspect ratio, and they experienced higher hydrodynamic forces and torques, and therefore their mobility would most likely be governed through straining. So that's something to also pay attention to. However, like much of how we're inferring that this is their movement through a soil profile, it is not truly representative about what is out there in nature. And this is actually part of what I study by looking at how soil microplastics of different types of morphology, sizes, types deposit down the soil profile. And I do hope to share that information with you all sometime soon. Another factor that really does influence how microplastics move in the environment, around the environment, is surface chemistry. So essentially it's because the surface chemistry of microplastics really do, coupled with the environmental matrix that they're in, influences whether they're going to be aggregate, they're going to sediment or deposit, right? So, and one thing to note about surface chemistry too is that it's subject to change based on the environmental exposure of the microplastics. Specifically like how long it's been out there and how much weathering they have undergone. Degradation processes such as photooxidation can create these new functional groups by way of photogenerated radicals, which result in the embrittlement of the plastic surface, so cracking on the plastic surface, and then as a result, further fragmentation. And then, this results in, you having smaller microplastics that are being produced, and therefore a higher surface area to volume ratio, allowing for absorption of different biota and contaminants. So surface chemistry really does influence where specifically these microplastics end up, because it can in turn alter like the density of the microplastic and may result in sinking and or suspension dependent on the density profile at depth. And finally, another physical chemical property that we really do pay attention to a lot with microplastics is that they have low their densities, whether they're low or high, because some microplastics have low densities, which are, when we're talking about low densities, we're talking close to the density of water. This paired with their size prompts, say, heteroaggregation that may occur, say, provided that there are a lot of colloidal particles that are present in this particular system. And if they're pretty light and they end up just staying at the top of the water surface, much of their movement will be through advective transport. So a lot of it is just like through just being entrenched in currents or waves, as well as some kind of like wind-driven transport specifically. However, if they're heavier as a result of, say, having biofouling or having like different types of microbes colonize the surface of that particular plastic, among other things like heteroaggregation, increasing their density over time, provided that the density of the water column that they're in provided the density of the water column that they're in kind of allows, like based on the density profile at depth, they can sink and end up in the sediments. So taking this knowledge from colloid transport studies, primarily this is where we're leveraging a lot of our understanding about microplastic movement and such, has helped us infer why they're so ubiquitous in environmental matrices, coupled with a large amount of source material that is present. However, there needs to be things that convey them into these systems. And these different types of things that we see conveyed into these systems, particularly in soils, are in this really quick illustration that I created. Presently, based on the literature in soils, application of biosolids and sewage sludge have been shown to play a role in addition of microplastics into agroecosystems, particularly agricultural soils. So a study that was actually conducted approximately 18 years ago found that microfibers were actually a good marker to know if an area had a biosolid applied to it. So that is like how they are known historically to actually be big contributors of microplastics. Disintegration of plastic mulch has also been shown to be a major conveyor of microplastics into soil systems. Irrigation from collected rainwater and reclaimed water have been reported to play a role in adding microplastics in soil. However, this remains understudied and needs to be looked into further. Greywater irrigation and dry deposition have been shown to be conveyors of microplastics, similar to atmospheric deposition. And there is more amazing research that is coming out regarding how atmospheric deposition plays a role in conveyance of microplastics, specifically resulting in deposition of soils. There have been a bunch of studies that have come out from this research group in France, whereby they've looked at the proximity of an urban center and the surrounding soils around there, and have noticed that atmospheric deposition really does play a big role for entry of microplastics into soil systems. Another thing that remains understudied and is something that more and more research is coming out about is stormwater, especially stormwater conveyance from urban centers, because there's a lot of deposition of microplastics on impervious areas, which are pretty common within urban centers. And they've actually been identified as a major conveyor of microplastics into receiving waters and in turn sediment, leaving behind the big question of, however, is this true in soils? And this is actually what I presently study. And I can tell you anecdotally and hope to share my results with you all soon that, in fact, they do convey microplastics into soil systems. Once these microplastics are conveyed into these soil systems, they've been shown to move down the soil profile through bioturbation, specifically by burrowing biota like earthworms that select, interestingly enough, for less dense plastics. And so they move further down the soil profile. Tillage has also been shown to move microplastics a little bit further down the soil profile, as well as the consecutive wetting and drying of soils. One thing to note with the wetting and drying of soils, and the reason why I'm not saying rainfall specifically, is that this has been primarily studied in a lab setting and hasn't really been done in the field. They have not really studied how rain specifically does result in the addition of microplastics down a soil profile. So in sediments, so this diagram really just looks at plastic flows in general, from their manufacture and use, and like where they specifically end up. But much of how microplastics actually end up in our oceans, in our lakes and rivers, where our sediments are through through anthropogenic inputs. And what I mean by that is like proximity to land, proximity to cities, urban centers. That definitely is a big role in the conveyance of microplastics into sediments. Another way is actually wastewater treatment processes. Besides, the contribution of biosolids, wastewater treatment plants have been shown to actually have a small fraction of microplastics entering receiving waters. However, this is also dependent on the level of wastewater infrastructure and the efficiency of wastewater infrastructure you have present, to actually properly limit the amount of microplastics that are in our final in the final water that is released back into receiving waters. A lot of it actually comes from mishandled waste, and this can happen from, say, different activities such as tourist activities. Fishing, aquaculture, even though not noted here, are also contributors of plastic into the oceanic environment. So, looking at how they move in the environment, as well as the large source of material and conveyance, it is unsurprising that microplastics are ubiquitous and have been found across different environmental matrices, as well as have been found in products for human consumption, such as beer, drinking water, and salt. Microplastics have been found in mountain ranges. They've been found close to the peak of Mount Everest. They've been found in our oceans. I'm sure you're all somewhat familiar of the Great Pacific garbage patch. They've been found in our rivers, our lakes. They've been found in our soils. They've been found in glaciers. They've been found in our atmosphere, in stormwater, within organisms, in our food. Microplastics are everywhere. And the question is, like, they're somewhat inert. Like, there's this assumption that they're these inert entities and we shouldn't worry about them. But really, there is good reason as to why we should worry about them. So let's consider the production of a tire specifically. As oil and coal is refined, a compound known as naphtha is produced, which with the addition of natural gas create the monomers known as styrene and isopropene needed to create synthetic rubber. Different additives are added to make the tire durable, malleable, and usable for a driving car. For example, carbon black, silica, plasticizers, textiles, sulfur, 6-PPD is a well-known antioxidant. And so when we drive said car, microplastics are produced. And as a reminder, these microplastics are essentially these larger plastics, just smaller, and they too come with all of these additives and chemicals. Some of these additives, just like the plastic item itself, can also transform when they're in the environment and create harmful byproducts, which in turn can end up in our environments and can have like some serious effects. And to talk more about this one, I'm actually specifically thinking about this study about coho salmon in the Pacific Northwest. So in the US Pacific Northwest, there were these mass die-offs of coho salmon when these adult salmon were migrating to creeks for many years that are now largely urban to reproduce. So folks were kind of perplexed with what could potentially be resulting in these die-offs when these salmon have been migrating here in years past. It turns out these creeks were not far off from roadways. So as we drive our vehicles, microplastics shed onto the roadway, and these small pieces of plastics, these microplastics, are then washed away by the stormwater into these creeks. And we know the Pacific Northwest has quite a bit of rain. And with them, they brought a byproduct of a compound known as 6-PPD quinone, which occurs when 6-PPD reacts with ozone. So as a reminder, 6-PPD is an antioxidant that's used in tire rubber so that it doesn't break down as quickly. However, when it reacted with the ozone, you ended up having 6-PPD quinone that was incredibly toxic at such low concentrations to these coho salmon, resulting in die-offs. Another way that plastics do pose a threat to our ecosystems is the fact that not only can they have additives that reach out, but also can act as a vector of pollutants, such as pesticides and heavy metals. A great example of this, actually, a recent investigation has looked at how surface roughness specifically, which is something that does happen to plastics as they're out there in the environment, that they weather and they become rougher. Surface roughness does result in like absorption of contaminants, like a higher affinity of organic pollutants specifically. It's been shown to carry pesticides, to have pesticides and heavy metals absorb onto them with ease. Mechanistically, what this can look like is you have microplastics that are contributors of these chemicals that may leach out and harm organisms that ingest them, and also the fact that they act as vectors for different types of pollutants that they may come across. And as I mentioned, they're pretty ubiquitous. Say you have plankton consume these microplastics, and then a small fish like anchovies, for example, consume these microplastics that have these toxicants sorbed onto them, or don't have toxicants, but do have these additives in them, right? And so that eventually ends up desorbing, and then you have your larger fish eating the smaller fish, and then we eat said fish, or we eat the smaller fish. So the potential for biomagnification of different types of contaminants is actually pretty high with microplastics specifically. There are also direct ways too that we can end up consuming microplastics, and there's a lot of research that is actually emerging around how this affects human health, and that is through, say, salt, for example, which has been identified to that different types of microplastics from different sea salts from around the world were evaluated, and they had quite a bit of microplastics in them, as well as drinking water is something that has also been looked at pretty recently about how they too are contributors of us directly consuming microplastics. So there's a lot of research that's coming out in that realm, and I'll just say keep your eyes peeled. I cannot say definitively that this has human health impacts, but I will say that there is a lot of emerging research that might show us that they indeed do. So when we're talking about effects, however, in soils and sediments, it's somewhat different. It isn't really like talked about or hasn't been looked at in terms of like the food web level, which it really should be. And I think it's because a lot of the research around soils and sediments and microplastics were actually not that dominant until 2018, 2017, 2018, that's when people were like, oh yeah, we should really look at microplastics in these like solid matrices. So with soils, they have been shown, and this is in no way exhaustive, there's quite a bit of research that is emerging around effects. And soils have been shown to actually change the soil biophysical environment. A really great illustration by Machado et al., published in 2018, saw that depending on the different types of plastic, you had different effects. So for example, with fibers, polyester fibers, for example, they saw an increase in water holding capacity. They saw decreases in soil bulk density with polyester fibers, decrease in soil microbial activity, soil structure and function. There's been like some kind of functional change. So depending on the different types of microplastics, it yields different effects, and their morphology also yields different effects. Then there have also been changes. So there have been a lot of research looking at soil fauna specifically earthworms, trying to understand what happens to earthworms when they're exposed to microplastics of different sizes and different types. One thing that is notable is the changes in the mass of the worm itself. Specifically when they do have access to these microplastics within their burrows. There have been studies that have also seen that in soils that microplastics temporarily stimulate microbial activity, which makes a lot of sense because they do leach out a little bit of carbon. However, because of the CN ratio of these plastics, eventually these microbes undergo nitrogen microbial starvation and can't really actually effectively degrade or be happy, I guess, on the surface of these microplastics. In sediments, specifically, what has been looked at is the microbial communities. And, sediments are really such an important facet of our global biogeochemical cycling. And the presence of these microplastics have actually shown a shift in the microbial communities that are present there. Much of that is attributed to potentially it could be the type of things that are leaching out, of the type of additives that are leaching out from these microplastics, those that successfully colonize those microplastics versus those that don't, among other things. But now comes the question, how do we actually evaluate microplastics in soil and sediment? So in soils and sediments specifically, this is a really great illustration from, if you're interested in microplastics in terrestrial environments, I highly recommend reading this chapter book. How we kind of sample it in soils and sediments is we core. That's what we do. We core our soils, we core our sediment. And then shortly after, what we do after collecting our cores is we sieve them. We sieve them because uniformly doing a density separation in a uniform sample is much better, is simpler, I suppose, than doing it in a somewhat incredibly heterogeneous sample, but also at the same time allows us to isolate what isn't really a microplastic, which is anything that is above 5 millimeters in size. So typically what follows after the sieving step is a density extraction using salt solutions such as sodium chloride, zinc chloride, sodium iodide, and calcium chloride. If you do soil science, sodium polytungstate is something that is desirable when it comes to fractionation. However, it's incredibly expensive and you have to use really large volumes depending on the size of the sample that you collect. But we do collect quite a bit of soil. You will have to use a large amount of volumes in order to effectively extract or do a density separation of these microplastics from these solid matrices. And then shortly after, what typically follows with that, and a density separation can take between 24 to 48 hours. Typically what follows after that is, doing a filtration of whatever floats to the top. And then one big issue though, however, is if you don't get microplastics in your initial density separation, a lot of it is somewhat occluded in this highly rich in organic matter solid matrix. So a digestion step is typically employed, such as Fenton's reagent, which is like the most cost-effective approach that's been used, or enzymatic digestion, which is incredibly expensive to use. So those are the two like methods that are primarily used. In the past, at the beginning of the creation of like these types of ways to evaluate and extract microplastics, there were base digest using like really strong bases to digest, as well as using acids to digest. However, much of the polymer itself underwent quite a bit of morphological changes, which made plastic identification really difficult. However, not to say that, say, using Fenton's reagent, for example, is gonna result in you having your plastics unaffected. No, they do undergo some kind of morphological changes on the plastic, which makes identification a little bit more difficult, but it's kind of viewed as like a necessary evil. Recently, using more mechanical processes, however, have been coming to the center stage of extracting microplastics from solid matrices, such as ultrasonication of the sample after an initial density separation before you subject it to a second density separation. And that is actually what I do for my work. Recently, one thing I do want to note is that these methods are typically tested, however, on one type of soil. They've been tested on numerous types of plastics, but typically when these kind of evaluations occur, it's always done on a single type of soil or sediment, when we all know soils and sediments are diverse, and methods should be developed with that in mind. Presently, the state of California conducted a challenge experiment, which I participated in, for the extraction of microplastics from sediments in hopes to standardize the methods. So hopefully we'll see what that looks like across different labs who employ different types of methods. But after we extract them, how do we know that they are a microplastic? Because they could be like algae, for example. So we typically use the stereo microscope. We look for morphologies and colors like these, and we record their color and morphology so that we may establish a subsampling approach, largely because you will not be able to process all of the samples. When I tell you that a lot of our samples have a lot of plastics, you need to be able to come up with like a subsampling, like representative approach. So one good way to be able to discern whether something is organic or not is through stereomicroscopy. And then once you're able to kind of categorize it based on color and morphology, we can come up with a really great approach to be like, okay, you need to at least, you know, for your subsampling approach, not to say that this is everyone's subsampling approach, but it could be like the first 20 particles of one color and one morphology type. That is what is going to be subsampled there. So it has to be for each color and this number of particles and this type of morphology. So this is just an example of what we use as a key to be able to identify what it is specifically that we're looking for. So fibers obviously look like fibers. They can be fiber bundles, fragments, or anything that are not uniform-like in size like this, so like a fiber, so that is a fragment. Spheres are very easy to identify. Foams look like essentially foamy little pieces. Think of styrofoam, but like at a very small size, very small size styrofoam. And then we have films as well, which a lot of it actually comes from food packaging. So you've got your microplastics, or so you say, what you think are your microplastics, right? You need to be able to actually identify the polymer. You need to confirm that what it is you're looking at is truly a microplastic, not some other type of organic like entity that's out there. So one thing to note about a lot of these, with the exception of these two here, your samples need to be relatively clean in order to actually identify polymers with confidence. And that's why we do the extraction approach, the isolation approach when it comes to microplastic, trying to evaluate microplastics in, say, solid matrices. FTIR and Raman microscopy specifically are the most commonly used ways to identify microplastics, which is great. However, we're often met with size limitations for evaluating the microplastics, with 50 microns for FTIR microscopes and 10 to 20 microns for Raman. Another thing too with FTIR, they're not really the best at identifying cellulosic fibers or microplastics, whereas ramen is better for that. But one thing to also note with ramen is that you're having like a really brightly colored microplastic can cause some potential hindrances. One thing that is also not noted using FTIR or ramen is that they can't help you identify the kinds of additives as well as additives that are in these plastics. So therefore you can't really effectively say that this is the kind of effect it might have in whatever matrix that you found it in. Additionally, a lot of the polymer ID databases are for pristine materials. So with FTIR and ramen, you get a spectra. And what you typically do is you try and match the spectra based on spectral libraries that have been established by different labs, among other things, among other like labs and also like materials or material scientists, among other entities, right? However, one thing to note about specifically these spectral libraries is that these databases these libraries are for pristine materials, not materials out there in the environment. So confirming polymer ID can be very difficult, and that is why we need, open source software or spectral libraries of environmental microplastics. And what we mean by that is like microplastics that have been out there in the environment for a while, that have been isolated from studies. So there's this awesome spectral library that does just that, and that is OpenSpeccy. And they have been helpful in like helping us confirm what we found is actually, say, polyethylene. It may somewhat look like it when you look at the spectra, but having a spectral library to actually match this to confidently is always a great way to move forward. There are more destructive methods employed that can help us identify the polymer and their additives. However, you just receive a concentration range in mass and not numbers. So you don't really like understand the magnitude. You only understand it from a concentration base, when we kind of have to think about microplastics from mass and count, like how much is out there. However, yeah, so with these specifically, you have to choose to isolate and identify what you had first. However, like that is the big issue with pyrolysis GCMS or TGA, FDR, and TGAMS, which essentially burn your sample and are able to tell you how much there is that there is in that sample. The thing with them is that if you don't do the extraction step, the isolation steps, you really don't have access to knowing specifically what type of microplastics are in there. And when I talk about type, I mean like the characteristics of the microplastics that you have present in your sample. So typically it's encouraged that you do that step of density separation and isolation prior to using destructive methods. However, you'd need to be able to standardize and ensure that whatever it is that you're adding for density separation can be kind of canceled out in the background. So thermoanalytical methods like TGA, FTIR, and TGAMS, for example, work well when you have a known matrix and known sample, but not really for unknown samples. And so that's something to also pay attention to, that there needs to be a continuous effort to come up with like standardized ways to optimize whatever it is specifically you're using to identify your polymer efficiently and effectively. So out of the destructive methods, what is actually preferred is pyrolysis GCMS, because it really does help allow you to understand the concentration of the plastic that you have in your matrix, as well as any type of additives it may have as well. So lastly, I like to kind of end on a hopeful note about how we can effectively mitigate microplastic pollution. And one thing I really do want to emphasize is that it requires a multi-tiered approach at the community, industry, and legislative level. Plastics are such a big part of our built environment that it's nearly impossible to avoid them. For some of us, it is such a crucial component of our lives, such as the use of straws, and that is something we should acknowledge and respect. If we have the means to divest from plastic, we can try and limit our use of it by making substitutions such as, say, a reusable water bottle. However, I want to acknowledge this solution can be a privileged one and can be made possible by having water infrastructure you can trust. This applies to all plastic substitution approaches. And one thing to note about that water infrastructure point is that there also needs to be infrastructural changes for us to be able to divest from the use of plastic in our daily lives. At the same time, I think it's important that as consumers that we educate ourselves and others about the effects of what we consume, and where what we use specifically ends up is important to making conscious decisions within your capability to do so. One other way that has been really helpful in resulting in like effective mitigation of microplastic pollution is that of better legislation. So I think expressing concern to your policymakers around microplastics or plastic pollution as a whole is incredibly important, as well as advocating for better waste management, enforcing of responsibility of industry, specifically when they produce these types of materials, like where do they end up, how do they end up affecting our environmental health, our human health, is something that needs to be brought to the forefront. The state of California is leading the charge in this specifically, whereby we had legislation that was recently passed around identifying microplastics in drinking water's like effects on our human health, and how we can evaluate and understand the concentration of microplastics in our drinking water, as well as how we can minimize the effects on our coastal ecosystems. So that is something that the state of California has been leading the charge of, and I think that if we did that nationally, it could be pretty effective. And then lastly, industry people should make a conscious effort to create materials that have limited effects to human and environmental health, and should be held accountable in the innovation of these materials presently and in the future through extended producer responsibility. Yeah, and so that pretty much concludes my talk on the state of the science of microplastics out there in our environment, and I'm happy to take any questions.